That's it. Marisa has golden hands. She fixed. Oh, yes. Yes. So she fixed it. When I touched it, everything started falling apart. So, anyway, let's finish with electron transport chain in sulfidrogens, and then we will move to nitrogen cycle, and that will be our final lecture. And then I will record last lecture and the one that I didn't record in post. And I want to say Instagram, not Instagram. Eclat. Speaking about that, you guys don't have a group for Micro 311? No? Not like a Discord group chat, no? Because I found out that Bio 322 does have. So yeah, I didn't know. But apparently, I mean, I joined, and because I didn't know that I cannot change my name, I actually joined with the right name. So I just didn't know. Nobody told me. I thought that you have to put your right name, and I put my right name. My Marina Lazic, and that's what I put there. Like, why would I call myself, I don't know, Sunflower when I'm Marina Lazic? That's anyway. Now I am logged in on group, so I can see what students talk about. Obviously, nobody wants to talk when they know I am there. Big shock. But anyway, hydrogenase is the first one in the electron transport chain. It's the same enzyme, same type of the enzyme bifurcation enzyme that we talked about when we talked about methanogens and acetogens. And the enzyme, so all that reaction of hydrogenase oxidizing hydrogen has 8 electrons released. So it's quite intensive. They travel to cytochrome. And from cytochrome C3, which is typical cytochrome, electrons travel to some membrane-associated protein, which you didn't hear about till now. It's very unique for sulfidrogens. It's called HMC. And this HMC is, of course, membrane protein that deliver radicals, the enzyme that completes final reduction of sulfate to sulfide, H2S. So which are those two enzymes that will use electrons that are delivered by this spanning HMC protein? So there are two important enzymes that you will have to know: APS reductase and sulfate reductase. And APS is forming sulfate from sulfide, from SO3 to SO4, and sulfite reductase will, well, this should be SO. From SO4 you get SO3, and from SO3 you get H2S. Sorry, it's good. I didn't make a mistake. I know that with those numbers and words it can be quite confusing, but the point is that this is sulfide. So hydrogen would be electron donor, SO4 2 minus or sulfate is electron acceptor. First reduction goes from SO4, we will get SO3, one of the minus done by APS. Then when you have SO3 minus sulfide, then comes the enzyme sulfide reductase that will reduce SO3 2 minus and make H2S, which is the same thing. Again, the name says sulfide reductase, so you need to have sulfide to be reduced. One interesting thing about sulfate reducers or SRBs is that these organisms are causing corrosions on bridges. And this is, I like to ask this question in all my classes because I found it easy to memorize. They often can consume oxygen at the metal surface, like anaerobic microbes. And then that metal surface becomes anaerobic or oxygen depleted, again, thanks to microbes. And when that oxygen depleting area with sulfidrogens or sulfate reducing bacteria will occupy this oxygen depleting area because they are anaerobic, so they just like to live when there is no oxygen. And when they respire that sulfate available in our oxygen depleting area, they can create hydrogen sulfide. Where is the problem? The problem is that this hydrogen sulfide, which is thereby metabolic byproduct, react with metal on bridges, and that creates metal sulfides, and metal sulfides can create corrosions. So in order to avoid corrosions on bridges, they do like a steel coating to prevent microbial biofilm formations on the bridges. Another thing interaction, again, which I really like to ask, is symbiotic interaction between syntrophic interaction between anime and sulfate-reducing bacteria. ANME is a new group of organisms. Did somebody ever heard about ANME? Yes, I can see bio 322 people because that's what we talked about that a lot. And there is a big confusion, which I'm hoping I will clarify now if I didn't earlier. Where is the, like, what are ANME? ANME stands for anaerobic methane oxidation. So literally, ANME means anaerobic methane oxidation. So, and these ANME, organisms that perform anaerobic methane oxidation, initially they were discovered as methanogens, like organisms that make methane. We knew that they have methyl coenzyme A reductase. We knew that they can make methane, so we would assume they are methanogens. Now, later on, it was found out that these organisms live in syntrophic interaction with sulfate-reducing bacteria, and in that case, when they are with SRB, they actually oxidize methane anaerobically. So first of all, it was weird because we knew they're methane. Second of all, it was weird because we always knew that methane oxidation is strictly aerobic, which means requires oxygen. Remember, you have to have oxygen for methanotrophy to oxidize methane. You have to have oxygen in terminal electron acceptor. You have to have oxygen to activate methane monooxygen. The thing with these organisms, they actually don't oxidize methane with methane monooxygen. They don't have same enzyme system. They're completely different, but they still oxidize methane when they are with SRBs. So that's one very weird and still not researched enough. Like, how is that happening? We still don't know, or I should say, I don't know. It is possible that somebody already published the paper or discovered. I don't know, honestly. I never researched it. I probably should, but I never. It's not my area, so I never have enough time. But it is interesting thing to. Maybe they did some metabolomics or proteomics or transcriptomics to figure out how is this actually happening. From now, we know that and all activity this between methane anaerobic oxidizing archaea. So instead of producing, they consume methane, but they are methanogens. They are discovered as methanogens, but they are actually anaerobic methanogens. So that was a tricky question that I had in biology which confused people. And I won't ask that question again because I realize that it can be it's not very straightforward, so it can cause confusion. But it's interesting. So what do they have? So ANME is consuming, so they are consuming methane, and it has to be participation of sulfate-reducing bacteria. Like, they cannot live one without each other. So that activity of anaerobic methane oxidation won't exist without SRBs. And from what I know, ANME have not been isolated independent. Like, they're not found in arsenic culture. They're always with SRBs. Terminal electron acceptor here is obviously sulfate, and sulfate-reducing bacteria are providing reducing equivalent. So even though it's not very energetically favorable reaction, it is occurring in syntrophy between ANME and SRB. So technically, you have two organisms doing methane sulfate oxidation reduction. So whenever we talk about the different metabolisms in methane, we always use this as a special, like, anaerobic methane oxidation. The reason why is because we do know that there are some anaerobic methane oxidizers ANME that even can respire methane, like iron or whatever is available there, like iron 3+, something that can accept electrons. And we talk about that as a special type of methanotrophy. It's not aerobic methanotrophy that I am working on, that we, like Marisa was saying, it's near methan. Now, what else I had here? Yes, so just a slight, like, brief reminder of how microbes drive sulfur cycle. So sulfur is present in all life forms, and the microbial part of sulfur cycle is very close interactions with chemical part of the cycles, and it makes total sense, especially because we know that microbes are chemists of the life. So when life form dies, there is desulfurylation, so like from organic molecules, there is a stripping of sulfur, then those sulfur forms get back assimilated into the cell. They're attaching sulfur, they're attaching to sulfur 420 APS, and that enables H2S formation. Then H2S can be a substrate for anoxic and even some oxic organisms. We talked about organisms sulfur oxidizing that use oxygen and consume hydrogen sulfide. Again, that's different than anoxic autotrophs. And this is a mini summary of some processes that we talked about last class that involve that are involved in sulfur cycle. Just again, reminder, you already heard about it. So hydrogen sulfide formed by sulfate-reducing bacteria. It interacts with metal minerals, with iron minerals. Iron sulfide interacts with elemental sulfur, and the period is the product. One interesting thing that we didn't note, that we didn't mention last time, is organic sulfur in the ocean. And there is a very long name for this DMSP compound, which I obviously didn't put because it's so it's enough called DMSP. What is interesting about it is that it's a metabolism can be used in global cooling, so that marine algae, like algae in ocean, will produce this DMSP, which is a sulfur compound, and the ocean microorganisms will degrade DMSP to DMS. So what that means is that you have DMS as electron donor. DMS can be the sulfur compound that can serve as electron donor in the ocean. And when DMS gets photo-oxidized, like oxidized at light, it creates aerosols alongside with like aerosols that interact with sulfur dioxide, sulfate, and metal sulfonate. And all these compounds can contribute to the global cooling, which we all need. So technically, microbes make that DMS compound that can help in global cooling. So it's an interesting thing that I don't know enough. I've never explored, but it is something to think about when you think about some research projects that you want to work on. Any questions before I move to nitrogen cycle? Yes, Rose.
What's the electron again? When the APS is of the
when APS is hydrogen. So all electron donor in this process has to be hydrogen. So you have hydrogen is the beginning of electron donor. We talk about sulfate reducing. We talk about sulfate reducing bacteria, and then you have APS, and you have sulfate. So that basically electron comes on hydrogen. And that's your first electron donor. Okay. Yeah. Okay. And sulfate obviously is like I said. Good practice. Are we going to move to the interesting part? Nice. I wish Brittany is here. She always loves this. Like, don't get like we are good, but Brittany is nitrogen person. We are more carbon people. My former TA really liked nitrogen, and every time when I would start this lecture, she would be like, "Yay!" They matter. Oh yeah. So you know Brittany, she's like big nitrogen person, and I will never forget her face when she would sit all in first row, and then when I'm giving me a nitrogen lecture, she's like, I need to see big smile on her face. It's literally like it's so enjoying. Okay. Anyway, nitrogen cycle. Let's talk about fun nitrogen species. It's not long, and I'm pretty sure you all remember glutamate dehydrogenase and glutamine synthase, and that isn't it the best metabolism, right? I don't know. It's like crazy to go like go to the therapy. No, it's actually very easy. Like after 10 years, when you actually figure once when you have like you have like a arousal, like yes, that's it. So 10 years I had no clue what that means. Now I figured it out. That happened to me, honestly. I won't tell you when that happened because I'm embarrassed. Yeah. So anyway, nitrogen. So when we talked about nitrogen cycle, this the picture of nitrogen cycle that you will see everywhere. Like we share this picture, like this is from Wikipedia. It's not too special. You can make this picture by yourself and you do it without reference. All these processes are interconverted, and we will go through each of them together. I mean, just looking at that, you can kind of so I always start like using the cycle. I like to start from nitrogen fixation because nitrogen is all around us. Like the nitrogen form is like here around us, but we cannot live on that nitrogen. We have to assimilate it somehow. However, organisms that have dinitrogenous complex, of course, can. And those are, as we know, diazotes. So nitrogen fixation would be the first step. But then next steps that are following that nitrogen fixation, because we do know that nitrogen fixation results in ammonium, NH3, then you will have something that will oxidize that ammonium, and that would be nitrification. Who is doing that are ammonia oxidizing bacteria and nitrate oxidizing bacteria. Include, and then also there are COMAMOX that are completing all processes of nitrification together. But then you have denitrification, which can be dissimilatory or assimilatory, which is, or can be anaerobic respiration. And then you have anamox, which is anaerobic ammonia oxidation. So it is nitrification, but it doesn't require oxygen. In fact, it's completely anaerobic. Who remembers what is anamox? I can, yeah, okay. I mean, you have PTSD of that, right? Yeah, okay. I know. I'm sorry. But I do want you to understand that there is also nitrification, just instead of NO3 minus, which is nitrate, it's nitrite, which is NO2 minus in anaerobic. So COMAMOX are also nitrification, and the people from MIT to know COMAMOX are doing what ammonia oxidizer said, nitrogen and NOB do together, and we will also nitrification. And all these organisms are running nitrogen cycle and interconvert nitrogen forms into the donors and acceptors, pretty much like in sulfur cycle. Nitrogen also has various oxidation states, which makes it suitable for metabolism. Now, why nitrogen cycle is important. So it all started back in the long 70s, in 1970, when those two guys, Haber and Bosch, discovered chemical process for nitrogen fixation. So that's when the massive production of chemical fertilizers started. Is there anyone who heard about Haber-Bosch process? So that, I'm glad, I'm good, I'm very happy that you remember that. And even though at the time it was, I think they got some Nobel Prize for that. Yeah, they did. That thing actually messed up us a lot. Like, even though at the moment it was great, like, oh my God, we can, improve food production, fantastic. Like, there are many hungry people in the world. Yes, but what happened afterwards? And this is what happened. Because the only organs that naturally does fixation of nitrogen are microbes. So this was chemical synthesis. But when that happened, when they actually invented this chemical synthesis, like artificial nitrogen fixation, that increased tremendously the use of synthetic fertilizers. What that means, like all those synthetic fertilizers are nitrogen-based. So that completely damaged ecosystems. Like humans completely disrupted nitrogen cycle, and nowadays we are actually responsible for vast majority of fixed nitrogen, more than microbes could ever do. But what will happen, and what is happening, what we see is happening in agriculture, there is that point, like we are reaching the point of nitrogen saturation, and then that ammonia, because most of them are ammonia-based fertilizers, and what happens is you're literally overloading a soil with ammonia. And one thing this is not in lecture notes, what happens when you overload your soil with synthetic fertilizer, especially ammonia, you have that overload on one side. On the other side, you have massive floods because of global warming, because of but you have creation of anaerobic, like minus oxygen, anoxic area in the soil. When you have anaerobic area in the soil, you have enough ammonia. Like salt is full of ammonia oxidizing bacteria because they are doing nitrification and nothing is good for plants. They are doing the production of nitrites, like alongside with nitrate oxidizing bacteria, when there is oxygen. The moment when you lose oxygen, the moment when area is microaerophilic, not completely anaerobic, but microaerophilic, these ammonia oxidizers shift their metabolism toward production, or so they don't do ammonia oxidation. They do something called nitrifier denitrification, which means they produce nitrous oxide. Nitrous oxide is 200 times more important than so I'm actually quite good. But when you look all dots and when you connect all metabolic dots, we are extremely endangered species. Like we, the problem is that like people often don't think about that. They say how they don't think about how hydrogen economy can affect global warming. Well, yeah, great, but hydrogen will rob atmosphere for hydroxyl radical, which means the only thing for methane, what are we going to do? We're going to melt. So again, this is another time when I really push for understanding metabolism. Like I don't think that everyone should be microbial physiologist, but everyone should understand the consequences. Not because we love science, but because we love life. Again, just my crazy thoughts about it. So many fertilizers in agriculture, many reactive nitrogen species that are showing up polluting water zone, nitrogen oxide in the atmosphere. Again, nitrous oxide, big problem, which I just told you about. So going about all of this, I think it's quite clear why we need to understand microbiology and microbial significance in the nitrogen cycle, because microbes do run all points of nitrogen cycle. We are just consumers, technically. Like, what do? All what we made are, like, when we die and we decompose, we release nitrogen species. There is nothing else that we do. Like, we don't make that. We don't fix nitrogen. We just consume. We make amino acids, but again, that also has to come from somewhere. So, and again, same like sulfur, nitrogen has various oxidation states, which obviously you don't have to know, same with for sulfur. But the reason for me showing you all these oxidation states is just to show you that nitrogen can participate in oxidoreduction processes and have various roles, pretty much same like sulfur. There are a large number of chemical reactions that include nitrogen and a large number of processes that include nitrogen. We will mention three main processes that will include nitrogen species, and those are nitrification, denitrification, and anamox. And in nitrification, we will have some comox, which will complete ammonia oxidation. So what is nitrification? Nitrification basically means oxidation of ammonia, NH3, and the model organism that we studied is Nitrosomonas europaea, but there are many other ammonia oxidizing bacteria that are very hard to grow. Their fun physiology, not fun to raise. It's like a teenage doctor. So nice when you grow it, but not help me. Yeah. So obligated chemolithotrophs, so that means they have very low biomass production. Litrozo is the prefix. Again, people from Bio 322 know that. For them, that's important. For you, not so much. Oxidation from ammonia to nitrite. So nitrite is NO2 minus. Nitrate is NO2. Ammonia oxidizing bacteria doing first step nitrification, which is oxidizing ammonia, blah blah blah, to nitrite. Then another group of organisms come, nitrate oxidizing bacteria or NP, who take this nitrite, oxidize it, and use and make nitrate. Now, and this is pretty much what I just said. Ammonia oxidizing bacteria, oxidation of ammonia to nitrite, and they live very close to this NOB because NOB take nitrite and make nitrate. So they complete nitrification process. So AOB plus NOB full nitrification from ammonia to nitrite, from nitrite to nitrate. They're both chemolithotrophs, which means they use inorganic source of energy. Source of energy for AOB is ammonia, source of energy for NOB is nitrite, which is NO2 minus. They both fix carbon dioxide with Calvin cycle, which means they need a lot of energy, which means they have to run that reverse or cyclic electron flow to just generate more and more proton motive force, because they're just so limited for proton motive force. These two groups of bacteria, AOB and NOB, are literally splitting process of nitrification. They are taking fixed nitrogen into the ammonia, so the azotrophs will fix nitrogen into ammonia, and these AOBs and NOBs will control how much ammonia will be converted and oxidized to different forms of nitrite and nitrate. So again, they are interconnected. They are very famous by causing nitrogen pollution, because when they are in microaerophilic environment, AOBs actually, they are producing nitrous oxide. They can also, one interesting fact is they can also use urea as a substrate, and they break urea to ammonia and carbon dioxide, and then they use that ammonia and convert it to nitrite. So again, they are very famous by that. They have, not all organisms have urease. Ammonia oxidation is very interesting from physiological standpoint. And there is a very good evolutionary connection with methane oxidizing bacteria, which again, probably people from biophysics already know. In the wastewater treatment, because it's full of ammonia, it's very common to use ammonia oxidizing organisms. So you will find them a lot in soil. While in the oceans, you have the third group of organisms that participates in nitrification, the group of organisms that was discovered in 2005 called ammonia oxidizing bacteria. And they're interesting from a few standpoints, especially because they have way higher affinity for ammonia. So in the process of oxidation, ammonia is the electron source, like energy donor, electron donor, energy source. And ammonia is oxidized to hydroxylamine by enzyme called ammonia monooxygenase, or AMO. Then hydroxylamine, this compound NH2O, NH2OH, is oxidized by hydroxylamine dehydrogenase, or hydroxylamine oxidoreductase, and it's oxidized to NO, which is nitric oxide. And then nitric oxide is converted to nitrite, NO2 minus, through unknown enzyme. It's still not known. But the point is, you go from ammonia to hydroxylamine, from hydroxylamine to NO, from NO to NO, and then NOB. So the most important enzyme in ammonia oxidation is ammonia monooxygenase, or AMO, and that is the powerhouse of the cell. It's an ergonic process because electrons have to be delivered to the enzyme to be activated, pretty much similar like methane monooxygenase that we talked about when we talked about methane oxidation. So MMO, methane monooxygenase, and AMO are evolutionary strictly related organisms, strictly related and evolutionary connected enzymes. They even have like a similar, like same enzyme, like they have same structure. They both require copper as a cofactor. And I'm talking about particulate methane monooxygenase, because methane oxidation can be performed by soluble enzymes, which we didn't talk about. But particulate methane monooxygenase is quite similar to AMO, which means methane monooxygenase will recognize ammonia as substitute oxide. What that means for metanotrophs is that they get poisoned with hydroxylamine, unless they have hydroxylamine oxide, but that's a whole different story. So, but the point is that have to be activated by oxygen. It requires oxygen as a terminal electron acceptor and to activate AMOs. And so, absolutely required. That's another thing. Again, that other enzyme that oxidizes methane requires completely different cofactor, requires iron, not the same like AMO. One oxygen atom is used to create hydroxylamine. And if you remember methane oxidation, one oxygen atom is used to create methanol. So if you see chemical formula, methanol would be CH3OH, and this is NH2OH. So it's just structurally similar. The same how methane is CH4, ammonium is NH3. So very similar structural chemistry. Reductants come from quinone pool. In methane oxidation, that's quite different. So reductants come from different sources in different parameters, so that's not the same. Another step of ammonia oxidation is hydroxylamine oxidation to nitric oxide. And this dehydrogenase oxidizes hydroxylamine in periplasm, and then electrons are released and nitric oxide is created. So what happens with those electrons now from hydroxylamine oxidation? And this is quite important. So released electrons, so electrons that originate from hydroxylamine oxidation, will travel to this specific type of cytochromes called C554, and there is another one called C552. There are two cytochromes. I mentioned only one here, that will be enough. And there is important reason why I mentioned that. So parts of those reductants will go to support ammonia oxidation, while the other part will travel to quinones to contribute to the PMF. And the part of electron transport chain are typical cytochromes that pump proton and create PMF. Now, the reason why I'm mentioning these cytochromes, like 554 and 552, which I didn't put here, metanotrophs, even though we say that they have similar enzymes and they're actually emotionally same, metanotrophs can oxidize ammonia, but because what people would think that metanotrophs would be great liquefiers and they could live on ammonia as energy source for ammonia. Why they can? So they can use, they can oxidize ammonia and utilize it as nitrogen source, and they will make hydroxylamine. It won't kill them because they evolved hydroxylamine dehydrogenase that will detoxify hydroxylamine alcohol. But metanotrophs do not have this cytochrome, 554 and 553, which means they have nothing to deliver electrons that originate from hydroxylamine oxidation to the electron transport chain, which means they can't survive on ammonia as a nitrogen, as an energy source. They have to have met. All right. Cyclic electron flow is what they have to run to create NADHs because they fix carbon dioxide, and carbon dioxide need a lot of proton motive force, so partially reductants have to be back to AMO. And the only way to create NADH for carbon fixation is to run that cyclic electron flow, like reverse electron flow, that creates additional, that spends additional proton motive force. So cyclic. It's simple, that's the only way, because quinone is not enough electronegative to donate red. Ammonia oxidizing adhere, which I mentioned at the beginning, I don't know much about them. I know that they're very young organisms, like from 2005. They are powerful ammonia oxidizers, and they run nitrogen cycle in the ocean. They have different pathway than ammonia oxidizing bacteria, and I'm not covering that here. It's similar but different. There is enough difference that can complicate your understanding. One thing that I do want you to know from them is that ammonia oxidizing adhere have ammonia monooxygenase that has much higher affinity for ammonia than ammonia oxidizing bacteria, which means they will send much lower amount of nitrogen of ammonia in the environment. So their Km is very low, which means they have very high affinity for ammonia. Second part of ammonia oxidation is part done by nitrite oxidizing bacteria, which means converting NO2- to NO3-. These organisms are even more oligotrophic than AOB. They live in very close association, simply because the AOB makes what they need to oxidize. So they need NO2- and the AOB makes them. So they're like partners. They help each other. Well, actually, ammonia oxidizing bacteria help NOBs. There is no other way around. They are very aerobic, sensitive to high oxygen levels. So, but they need oxygen, but they are sensitive to high oxygen level. The redox difference between nitrite and nitrate is not big, which means it's very short electron transport chain. It's only one cytochrome involved. There is nothing too special. It's very short pathway, which again means, as you know, they're very oligotrophic. There is not much biomass. So they literally produce nothing. This is how. So again, the electron transport chain is quite short and there is nothing too special. The only thing you never heard about is this NXR enzyme that oxidizes NO2 minus to NO3 minus, and electrons that are extracted, they travel to cytochrome. Cytochrome is you already heard about. And from cytochrome, they go to the terminal oxidase, which is cytochrome AA3. Pretty much something that you already heard about. And there is a pumping of protons and proton multiforce only here at the end, in the terminal oxidase. So it's quite low proton multiforce. Very, very small. So they are very, very limited into the biomass production. They must oxidize tons of nitrites to be able to grow, and they fix carbon dioxide, which means they need a lot of proton multiforce. But majority of proton multiforce is used for this cyclic electron flow because they have to run cyclic electron flow to create NADH, because NADH is what carbon cycle to fix carbon dioxide. So they are very energetically limited, which you can conclude based on the shortness of their electron transport chain. Only one enzyme, 2 cycles. That's it. So same way how to complete nitrification process, you need AOB and NOB. The organisms that can do complete ammonia oxidation are called COMAMOX, and the COMAMOX stands for complete ammonia oxidation. They're even younger than ammonia oxidating bacteria. They were discovered in 2017, and they're also nitrifiers, so they have capacities to oxidize ammonia to nitrite and nitrite to nitrate. So pretty much like AOB plus NOB together. So, and they are apparently very efficient ammonia oxidizers. The representative of the group is the organism called Nitrospira. It's a very good organism in the oxidation of nitrite to nitrate, but it does require ammonia as a primary energy source. So COMAMOX has the highest affinity, the highest affinity for ammonia is what COMAMOX has. So AOB are the lowest, like they need a lot of ammonia. Then AOA, then COMAMOX. So COMAMOX have even higher affinity for ammonia, which means low Km. So they're better than ammonia oxidizing RTF. So they're literally capable of picking up all available ammonia. So if you could enrich them, which some people did, they can do nitrogen transformation, which contributes to detoxification via radiation, so you could engineer that systems. And I'm really sure that Brittany was talking about fish tanks, which is really great way of, again, you already heard about that. So in fish tanks, you have excretion of ammonia waste that goes to microbial reactor. Nitrifiers oxidize ammonia to nitrite and nitrite to nitrate. That's what COMAMOX would do. And then water enriched with nitrate will go to support growing of plants like lettuce. And our lab, Brittany and Taylor, were doing that. There is a very cool poster on 5th floor if you want to see how that looks. That's what Brittany and Taylor were doing. Anyway, so moving on to denitrification. So we covered nitrification, IAOB, NOB. What about denitrification? So dissimilatory respiration of nitrogen oxides. And there are a few processes: respiratory denitrification, nitrate respiration, and eukaryotic denitrifier. So nitrate respiration and respiratory denitrification are commonly mistaken. So pay attention. That's something that people get very confused. So what is nitrate respiration? So nitrate respiration is alternative form of energy production in organisms like E. coli when there is not enough oxygen. So it is one branch of electron transport chain where nitrate is alternative electron receptor. You probably remember that from the beginning. Ubiquinone is now menaquinone. So again, instead of oxygen at the end of electron transport chain, there is NO3 minus. And there is no ubiquinone, there is menaquinone, and there is different cytochrome type. And the final enzyme is not oxidase, it's nitrite reductase, and there is much less proton multiforce formed. And the product of nitrate respiration is nitrite. This is respiration of alternative terminal electron acceptance. And we talked about that when we talked at the beginning, but I forgot which unit exactly, but you change the type, the cytochrome, you change the type of the quinone, and it's not oxidase, it's terminal reactive. So this is one branch of electron transport chain in capital. So this is not, this is nitrate respiration. So to survive nitrate respiration, cells have to have something for nitrite because nitrite is toxic. So usually they contain some reductases and they perform dissimilatory nitrite reduction just to detoxify the produced NO2 minus. Those reductase enzymes contain molybdenum as a cofactor, pretty old time. So that was respiration done by E. coli. What is respiratory denitrification? So this is a process run by various reductases that convert various forms of nitrogen into another form of nitrogen. So you start from nitrate. So you have nitrate reductase that reduce nitrate to nitrite. Then you have nitrite reductase that reduce nitrite to nitric oxide. Then you have nitric oxide reductase that reduce nitric oxide to nitrous oxide. And very often it stops here. But some organisms have nitrous oxide reductase that will reduce nitrous oxide to dinitrogen. So pretty much the name of the reductase fits with the substrate. So it's not really so hard. Nitrous oxide is very often where everything stops. Rarely there is nitrous oxide reductase. So respiratory denitrifying organisms alongside ammonia oxidizing bacteria make nitrous oxide greenhouse gas when there is no oxygen available because N2 production is not always happening. So you can imagine that getting better nitrous oxide reductase can really be something to think about in future because that will help detoxifying nitrous oxide produced. The example of respiratory denitrification is Pseudomonas. You don't have to know the name of the organism. I really don't care about that. The electron donor here is organic molecule, and it's oxidized. Organic molecule is oxidized, and in addition to organic molecule, which is usually carbon source and contributes to the electron donation, there is NADH involved. So this is typical like electron transport chain, that's beginning of electron transport chain. Nothing is really too specific here. So you have the electron transport chain that starts with NADH dehydrogenase, and then the electrons move to various carriers like quinone, cytochromes, and they are delivered to nitrate reductase, and then nitrate is reduced to nitrite. And then there is a next electron coming, reduce nitrite to nitric oxide, and so on and so on. There is involvement of other cytochromes that help in transporting these electrons. There is nothing really too special about this electron transport chain and nothing too hard. All what you need to understand is that respiratory denitrification and nitrate respiration are not the same. Yeah, what else? So again, this is literally what I just said. There are many electron carriers and reductases that contribute to the proton multiforce formation. And in summary of these denitrification enzymes, again, nitrite reductase and nitrate reductase. So every time will be reductase. So nitrite reductase reduce nitrite to nitric oxide. There are two types. Copper and iron, NIRK and NIRS. Nitric oxide reductase or NOR. It stands for nitrite oxide reductase. It's only one time copper oxidase, similar to terminal cytochrome oxidase. And the nitrous oxide reductase, which is very significant, not Z, again, nitric oxide, performs reduction of nitrous oxide to nitrite, so also copper. And you can see, and this is the only known nitrous oxide sink, and you can see that most of them are copper or iron contained. Eukaryotic denitrifiers are very interesting from evolutionary perspective. We talked about, you remember when we talked about sulfate and sulfidogens that live in plant and in trophozoan actually in that marine organism. So here you will have also fungi, but this is actually interesting fungi that has all these denitrification reductases, of course, were in mitochondria. So what's happening in cell, it starts from pyruvate. Again, this is eukaryote. Pyruvate gets oxidized to acetylcoenzyme A, which is converted to formate. Formate gets converted by formatic hydrogenase carbon dioxide. And electrons from that formate oxidation are used for nitrite reduction. And you get nitrous oxide production. So basically, all those reductases are there, like nitric oxide reductase. And at the same time, they also reduce oxygen. They reduce oxygen to water. So they do nitrite respiration and the oxygen respiration. That's hybrid respiration. Just it's nitrate respiration occurs also in mitochondria. So it's like a combination between two electron receptors in eukaryotes. This organism is, and then there is this one called benthic foraminifer, which completes denitrification to nitrogen, which is very weird. They do not have oxygen. Like in these sediments, there is no oxygen, it's anoptic. There is, and those benthic foraminifera still grow. And what people found is that nitrate was very abundant in those cells. And when they do labeling with radioactive nitrogen, they saw that NO3 minus was actually, so because they labeled nitrogen in nitrate, and then they saw that that labeled nitrogen appears in atmosphere in N2, which means that they actually use their own nitrites to produce atmospheric nitrogen. These foraminifera store a large amounts of nitrite in their cell, so it's very energy demanding. But what is weird is that they did not find any associating denitrified prokaryotes there. I can see mitochondria, so I would see it. Maybe they are not associated living, a single living prokaryotes, but mitochondria are bacteria, so they have mitochondria, and that's where this is happening. So that technically is a sort of like we all have symbiosis with prokaryotes. Special type of denitrification is that famous anamox or anaerobic ammonia oxidation. So anaerobic ammonia oxidation is using ammonia, and they oxidize ammonia to ammonium. So ammonia oxidized bacteria will use ammonia. Anamox will use ammonium. Ammonium is ammonia when it comes to the cell, and at the ultra pH becomes ammonium. So in order to be used in anamox, it has to be ammonium. Again, I was never very picky about that, but just FYI. So anamox utilize ammonium as electron donor, and terminal electron acceptor is nitrite, NO2 minus. That's why it's denitrification. So this is very energetically favorable reaction. And these guys are actually way better in wastewater treatment because it's an anaerobic environment in that wastewater, so they will thrive, unlike ammonia oxidizing bacteria that actually need oxygen. So they are used to remove ammonium from wastewater. Anamox couldn't work here because apparently this is too cold for them. But in warmer areas, they're used for bioremediation of nitrogen waste. They are planktomycetes. Planktomycetes are a group of bacterial phyla that we call in Bio 322 CPV, Chlamydia planktomycetes verrucomicrobia. One characteristic of planktomycetes is that they have compartments, like they have compartmentalized cytoplasm. And anamox are known by that because they have something called anamoxysomes, which are nowadays called true cellular organelles. And the anamoxysomes, they are surrounded by a special type of lipids called lateral lipids, which are very tight and extremely rigid. And these anamox are a place where those true organelles are places of ATP production where ammonium is oxidized to nitrite. Because and they fix carbon dioxide via Langdale, like Wood-Langdale or acetylcoenzyme A pathway, which is very energetically efficient. the reason why they evolved these lateral lipids in anamoxosomes, like they surrounded it in the cell, they made compartment, is because one of the products of ammonium oxidation and nitrite reduction is hydrazine, and hydrazine is very toxic. So they had to develop those lateral lipids to prevent leakage of toxic compounds into the surrounding environment. Again, which is just a summary of what I said. Ammonium donor, NO2 minus acceptor. Electrons from NO2 minus used to fix carbon dioxide. Because they use acetyl coenzyme A pathway, they make that acetyl coenzyme A useful for biosynthesis. It's typical Wood-1 tail pathway, same like methanogens and acetogens use. I already said that hydrazine toxic, that's a product of ammonium oxidation nitrate reduction. So this should be, it's not lateral, it's a lateral lipids. So LADDERAN, lateral lipids. Stupid PowerPoint converted it because he recognized it as a real word. It's not real word. It's lateral, not lateral. Please make that note. It's lateral lipids. And this is how those anamoxosome looks. So they're like a true compartment. How is that toxic compound produced? So there is an enzyme, of course, called hydrazine hydrolase. So they oxidize ammonium to hydroxylamine, and ammonium and hydroxylamine getting combined by enzyme hydrazine hydrolase, and they yield hydrazine, which is very toxic. And then there is enzyme hydrazine oxidase that oxidize hydrazine, and that release electrons, which are used to reduce NO2 minus. And proton monoforce is used to turn on ATP synthase that is located on anamoxosome membrane. So that they literally all electron transport chain happen in anamoxosomes. And this will be all that I had for this class. Next lecture will be recorded, and I will do my best to post it by Friday night. I will have to. And next Tuesday we have all of you presenting, and I'm looking forward hearing how that looks. Of course, then we will have preparation for midterm. I always give a review session, as you know, so I will lead you through that. Pretty much like I did for last year for Bio 322, now for midterm. So I will make presentation and I will lead you through, I will give you hints and points what will be on final exam. That doesn't mean you shouldn't study everything, like you should study everything, but I will take you through everything. I will give you hints, so please listen what am I saying.
Reminder for the presentations, for the syllabus, if you guys can all send me your presentations